Well, now that we've established what DNA is and where it is in our cells, it's now time for us to sort of have a look at the effects of what it does. And this is where we're going to start looking at specifically the different types of genes that we've talked about, alleles, and how they're passed on from one generation to the next. We're going to look at that by looking at the process of what we call inheritance, so how we inherit traits. And this is done through our simplest understanding of genetics, which is Mendelian genetics. So for this, we need to be able to define what a recessive dominant trait is. We also need to be able to define phenotype and genotype, heterozygous, homozygous, and sex-linked. We will actually look at sex-linked later on, rather than looking at it specifically in this video. We'll actually be looking at sex-linked in another video and looking at the diseases that are associated with that. So this allows us to be able to describe how a person is affected by the genetic trait, whether or not the characteristic is dominant or recessive, and to solve what we call monohybrid crosses, where we can work out the percentage or the proportion of the offspring that will be a particular phenotype. And then we will look at other problems for sex linked a little later on. So these are the key terms that we need to know, and you should add this to your definitions. You should be able to define these as we go through, and then we'll have a bit of practice with this terminology as well. So essentially, the basis of all genetics that we study at the moment is based on Gregor Mendel. He defined the idea that heredity is the transmission of traits from one generation to the next. Okay, and then these traits that we have, form the heredity or the genetic component of our cells, and then that in combination with the environment gives us the individual. Okay, so we're not all a pure product of our genetics. There's a lot of environmental factors that go into this as well. So genetics has grown up as the study of heredity and how traits are passed on. So this idea was actually, it's quite a long time ago that it was originally put together. This is before we even knew what DNA was. And Gregor Mendel discovered this by breeding pea plants in the garden of the monastery because he was a monk back in the 1850s. So in 1866, he argued that the parents pass their offspring on in discrete heritable traits, which retain their individuality generation after generation. So previous to that, it was considered that all our traits were a blending of the parents rather than individual traits being expressed or not expressed. So this is a look at sort of the pea plant experiment that Mendel did. So he looked at different plants, so their flower color, the flower position, seed color, seed shape, pod color, and stem length. So if you can see this is basically all the different types of observable traits of the plants that he was looking at. So I selected these 7 characteristics and then grew up different generations of these plants to see what would happen. Okay, so and then discovered that there must be some kind of gene that determines which of these options come out. So when we talk about alleles and different forms of a gene, we're talking about basically these decision points on the different traits that we see in an organism. As we've said before, these alleles are carried on our chromosomes, and we have the maternal chromosome and the paternal chromosome. Okay, they are homozygous, so they code for the same trait. But if we have a homozygous pairing of traits, okay, they must be the same versus a heterozygous. So you can see here that on this diagram, these traits here, which says this is an allele, so they're in the same locus, and we can see that they both have a capital A. So these ones would be homozygous. Okay, so these ones are homozygous because they would actually be the same. Whereas these ones here are heterozygous for an allele because they actually have a little b and a big B. And we use small and capital letters to define the difference between alleles to say whether or not they're homozygous or heterozygous. So again, we can see these ones would be homozygous, these ones as well. These ones here would be heterozygous, homozygous, as we go down. So when we refer to the genotype of an individual, okay, this is the allele combination, whether or not it is homozygous or heterozygous, and whether it is homozygous dominant, which means it would have two capital letters, or homozygous recessive, which means it would have two lowercase letters. And then if it's heterozygous, it has to have one of each. Okay, so we can see here that with blue eyes, this is a recessive homozygous trait. So you need two little b's in order to have blue eyes. Whereas brown eyes can either be a heterozygous trait, so big B, little b, or a homozygous big B, because the brown eyes trait, which we show with a big B, is dominant. So the term phenotype relates to the physical appearance. Okay, so I like to remember phenotype, physical, that same PH sound at the beginning of the word. And this is our observable characteristic. So in the case of our eye color example, it's whether or not the eyes are blue or brown. Okay, whereas the genotype is the letter combination, the BB or the little b, or the big B, big B. So when we refer to as a trait being dominant, this is the one that will be shown in the heterozygous individual. So if we have a big B, little b, it's this one. Okay, the big B would be the dominant trait, and that is the phenotype that we would see in the individual. Whereas the recessive trait would be the little b, and that's the one that is silenced by the presence of the dominant allele. Okay, so these traits remain hidden in the heterozygous individual. So we don't know if they have a blue eye gene or not because we don't see it in the individual. We would need to do a full genetic study in order to find that out. So when we do what we call Punnett squares and look at the breeding of individuals, we're looking at true breeding, so consistently producing offspring the same as the parents for a particular trait. So we would select parents that have round seeds in order to produce plants that have round seeds. And the effect of this we can see in dog breeds and cat breeds, different plants that have been bred over time to express specific traits that we desire as a society. So with this, we have P, which stands for the parent generation or the true breeding generation. And then we use these terms F1 and F2 to refer to the filial offspring generation. Okay, so these will have the combinations of the traits from the parental generation or the P generation. Okay, so these ones, we look at the traits that have been produced and then can work out what the parents were. The second filial generation is if we take two from here and breed those again, and we can see what traits turn up. We can look at this by doing a monohybrid cross, where we can look at breeding different allele combinations for the parents. So if we look at this one, we can look at our plant example again, and we have round and wrinkled, and we can see that the F1 generation gave round, and then the F2 generation gave round and wrinkled with a probability of 3:1. So when we look at what the generations provide or produce, we get an idea of what the genotype could be. Okay, so this is what Mendel did. He took specific traits, he bred them together with over about 28,000 pea plants. That's a lot of dedication. and found that he obtained similar results as predicted by his studies. So through this, he came up with a series of laws, and these are there are alternative versions of genes that account for variations in inherited characters. We call those alleles. Okay. For each character an organism inherits, there are two alleles, one from each parent. So this is where we can have our homozygous or heterozygous combinations of alleles. If the two alleles of an inherited pair differ, then one determines the organism's appearance, and it is called the dominant allele, okay, given the capital letter, and the other has no observable effect on the organism's appearance, okay, so it doesn't change the phenotype and is known as the recessive allele. The last one is the law of segregation, and this is that the sperm and the egg carry only one allele for each inherited trait. So this means this is getting back to them being haploid, okay, and that the allele pairs must separate during gamete production, which is what we have learnt in meiosis. So I'm going to end this here. Make sure that you have your definitions tied down, and in the next video we'll look at examples on how to draw Punnett.